Welcome to this e-module. This program is designed for the students of Bachelor of Science 3rd year in the subject of Botany, Semester 6, course code BOC 108, course title Cytogenetics and Plant Breeding. The title of this unit is Inbreeding Depression and Heterosis. Module name Heterosis and its application. I am Ms. Emma Fernandez, Assistant Professor in Bempe College of Arts and Science, Miramar, Goa. In this module, we shall see the introduction, general aspects of heterosis, causes, effects, and applications of heterosis. At the end of this module, the student will describe heterosis, state the general aspects of heterosis, list the effects of heterosis, explain the causes of heterosis, and at last summarize the applications of heterosis. Hybrid vigor is a hereditary process by which two genetically different individuals, when crossed with each other, produce F1 hybrids which are superior to the best parent or mean value of the two parents in respect to size, growth, yield, and fertility. The superiority of F1 hybrids over parents is referred to as hybrid vigor. It was G.H. Schill in 1914 who used the term heterosis for hybrid vigor. The term heterosis was used for the growth and vigor arising due to heterozygosity. Now let us see some of the general aspects of heterosis. Heterosis is governed by genetic factors. Hybrid vigor is a normal process in plants and animals, but in cross-pollinated plants it is more conspicuous than in self-pollinated plants. That means in cross-pollinated plants, the hybrid vigor can be clearly seen as compared to the self-pollinated ones. It is a natural process and occurs more automatically in heterozygous plants than in homozygous plants. Heterosis does not manifest equally in all hybrids of a species. Some hybrids show heterosis while others do not. Crosses between distinct type or genetically diverse variety exhibit greater heterosis than those involving closely related varieties. Heterosis shows maximum manifestation in F1 hybrids. In F2 and subsequent generation, it decreases gradually because the genes producing heterosis segregate. Heterosis is directly related to specific combining ability of the parents. It shows maximum manifestation in the hybrids of such parents which have got high specific combining abilities. In some agricultural crops, heterosis is large enough to be used in production of hybrid varieties. For example, in maize, jowar, bajra, wheat, and many more. Now let us see the causes of heterosis. Heterosis is basically caused because of two aspects: genetic causes and physiological causes. These causes are further understood or are they further explained with the help of hypothesis. Genetic causes can be understood with the help of dominant factor hypothesis and overdominance hypothesis. On the other hand, we have physiological causes which can be explained with the help of cytoplasmic nuclear interaction hypothesis and greater initial capital potential hypothesis. Now let us see the genetic causes in detail. Dominant factor hypothesis. In this, the increased vigor results due to large number of favorable dominant genes in the hybrid than parents. The crossbreeding generally results in the accumulation of the large number of dominant genes in the hybrid. For example, cross between A, capital B, small b, small c, and small a, capital B, and capital C gives a genotype of capital A, capital B, capital C. When all three dominant genes are favorable in the hybrid, it will show increased vigor than the parents. Coming to the overdominance hypothesis. In this, the heterozygote is superior to homozygote, and hybrid vigor increases with the increase in the amount in the heterozygosity. For example, heterozygous A1A2 is superior to A1A1 and A2A2. The genes A1 and A2 govern different functions. Thus, the heterozygote is more vigorous than the inbreds. Coming to the physiological causes. Cytoplasmic nuclear interaction hypothesis. According to this, heterosis might be caused by the interaction of cytoplasm and nucleus. It is due to the effect of changed nucleus and relatively unchanged cytoplasm upon each other. Greater initial capital potential hypothesis. In this, the hybrid vigor is due to an increased initial embryo size or due to so-called greater initial capital. Let us see the effects of heterosis. Heterosis results in the increase in the size of the plant. It increases the yield of the crop. The number and size of fruits and seeds increases. It increases the fertility of plants. It increases the vitality of the plants. It results in more efficient germination and growth. It induces early flowering and fruit setting. Now let us learn about one example of heterosis which is seen in corn. G.H. Schull in 1909 has shown that in corn, hybrids between inbreds of diverse parentage generally give greater hybrid vigor than that shown by hybrids between inbreds derived from same or similar open pollinated varieties. However, there was one problem. Most inbred lines were so infertile that they did not produce enough seeds for commercial planting. Schull's method was modified by a method called as double cross method. The double cross method for producing hybrid corn. Starting with 4 inbred lines A, B, C and D. So we have inbred A, inbred B, inbred C and inbred D. A single cross is made between A and B by growing the two lines together and removing the tassels from the line A so that A cannot self-fertilize and thus receive only B pollen. In another locality, the same method is followed for lines C and D, wherein single cross between inbred C and inbred D was carried out. The results of this were the yield of single cross hybrid seed is usually low because the inbred parent lacks vigor and producing small cobs. The plant that germinate from single cross seeds are usually vigorous, hybrids with large cobs and many kernels. However, it was undesirable for a single cross hybrid to self-fertilize as this inbreeding process commonly produces less vigorous progeny. Therefore, a double cross is made by using pollen from CD hybrid on the AB hybrid. So, a double cross was carried out between CB hybrid and AB hybrid which yielded a hybrid of ABCD, which had hybrid vigor of more higher than the hybrid CD and hybrid AB. Applications of heterosis. Heterosis has exploited at commercial scale both in plants as well as in animals. In plants, it is found more important in vegetatively propagated perennial plants, whereas in fruit and ornamental plants, if heterosis is once achieved, it may be maintained for a long time. Among cross-pollinated crops, heterosis is exploited in the form of hybrids, composites, and synthetic seeds. Intermediate phenotypes are preferred. In such a case, heterosis involves mating of parents having opposite phenotypes. The examples over here are general purpose cattle can be produced by beef type with a dairy type. The offspring commonly produced an intermediate yield of milk and hung up a fair carcass when slaughtered. Also, the same is true for the offspring from crossing an egg type such as a leghorn breed of chicken with a meat type such as the Cornish. Crossing phenotypic opposite may also be made to correct specific defects such as weedy relatives of agriculturally important crops may carry genes for resistance to specific diseases. Combining these two, we can get a hybrid from such a cross which may acquire disease resistance and successive rounds of selection combined with back crossing to the crop variety and eventually fix the gene or genes for disease resistance. These are my references. Thank you.